So as we have sequenced genomes and identified genes, mapped, annotated those genes, made connections between different genomes, right? Established which genes are homologous, both in paralogs and orthologs. One of the key things that has come out of this as we look at developmental genes is this remarkable correlation between the complexity of these important developmental genes and the organisms in which they are found. So, for instance, there are four main types of DNA binding genes that are found in transcription factors. The homeodomain is one of the most important ones. And in humans and other mammals, which are very, very complex, there are many of these. Other bilaterians, C. elegans, Drosophila, still have a good number, but not as many as the complex vertebrates. And then in simpler animals, we have a smaller number. And you can see it's not a complete correlation. It's not always one-to-one, but in general sense, the more complex you are, the more complex your toolkit genes are. Notice in the signaling molecules, there's a little bit of a trend also, right? So some of these important signaling molecules that can also be important toolkit genes, but primarily in these transcription factors. That's where we really see this major ramp up in complexity. So there's very clearly a general correlation between toolkit gene complexity and the complexity of organisms. But there are also some very, very specific increases that might even lead to, and there's, whether or not these are just really, really correlated or causative is a little tricky to figure out. There's some good evidence that these are actually one, if not the major driver of complexity for these specific examples we're going to talk about. And so let's look at a couple of trends, and then we'll actually put them onto a phylogeny so that you can see that. So some Hox genes and important control genes are found in the cnidarians, which are the simplest animals, one of the earliest branches among the divergence of the animals. And so that shows that these early toolkit genes, at least in a simplified form, were present in the very first animals. The earliest metazoans had these transcription factors, but they probably were not incredibly complex. And cnidarians and some other animals, ctenophores, I don't actually know if there's a lot of work done on them, but some of the early simpler animals have maintained this early kind of genetic simplicity, and that's also represented in their morphological complexity, which is nowhere near what we see in the other animals. So we can think of a primitive early simple toolkit in the first animals, and that some animals, including the cnidarians, have kept that kind of simplified form. Okay, so that ancestor probably had a very, very simple form. But as we move up the phylogeny, so this is a zoom in, but this would be the common ancestor of all metazoa, and then this would be the common ancestor of the cnidarians and the bilaterians. And once we get to this link in the bilaterians, we begin to see morphological complexity. And when we map this diversity of toolkit genes onto the phylogeny, we see a corresponding rapid evolution of the toolkit along this line that represents the ancestors of these complex bilaterians. So let's look at it. So we're going to focus in on that branch right here. So here's a more expanded view. Here we have the cnidaria with only 2 of these represented Hox genes. And I think this needs to be updated a bit, but the trend is very easy and clear to see. So notice cnidaria, very simple, few Hox genes. And then as soon as we get, and all of them really, but as soon as we get into the bilaterians, we have many, many, many Hox genes, sometimes dozens, right? But it's certainly at least 8, and other homeodomains are here also. Okay, so the Hox genes, there's this interesting thing. Now within this, there are complexities. So point number 1 is a diversification of all of the Hox genes in the ancestor of bilaterians. So that's listed there, the expansion of these Hox genes, especially the central ones. But notice that even the posterior ones and the anterior ones show some levels of increased complexity, but especially the central ones. But point 1, at the ancestor leading to the bilaterians, we have a dramatic increase in the diversity and number of Hox genes. Number 2, the posterior Hox genes become more complex in the chordates, right? So here we have this expansion of these posterior Hox genes rather dramatically, And there was actually a fairly minor expansion among a subset of the ecdysozoa, but really this is in, sorry, I said chordates earlier, but it to some extent, yeah, the chordates do have more of it, but it also applies, we could say in general, and they have it mapped here in the deuterostomes. So I'd be okay if you said it was in the chordates. That's fine because they have way more complexity than the echinoderms. But in general, we'll just say it's for the deuterostomes. And the deuterostomes have a more complex posterior section of the Hox complex than the rest of the animals. Okay, and then finally, in the vertebrates, we have this fourfold or tetraploid duplication of the entire Hox complex. So notice we have one line, which each of these color squares representing a different orthologous Hox gene. And then we have duplications, and they're generally color-coded: green for the central, orange and red, and yellow for the anterior, and then blue for the posterior. But the line represents a portion of a chromosome with all of the genes located on it, right? So we've got that there. And notice that for the vertebrates, we have 4 different lines. So it appears that this duplication was most likely a fourfold duplication of at least the chromosome upon which the Hox genes are found, maybe even the entire genome of an early vertebrate Hox complex. So notice the cephalochordates, right? Amphioxus only has a single Hox complex, although many genes in it. But vertebrates, including you and me, fish, amphibians, reptiles, mammals, all have 4 different Hox complexes found on 4 different chromosomes. And there have been loss of some genes, but we still have maintained many of them. And so we get this potential for increased morphological diversity and complexity of form. And so almost certainly these 3 events, the 3 that you need to know, led to the more levels of complexity and therefore much of the success of these groups of organisms. Okay, so just be aware of those. Again, know that there are 3 general areas. We're not going to get in that more detail, but just you can see it on the diagram here, and one of them, the expansion of the posterior Hox genes, refers particularly to one of these areas. The areas themselves are not really critical, but the concepts we talked about earlier are. And so these 3 key genomic events and expansions of the toolkit genes, particularly Hox genes, right? That's what we're focusing on here. But other toolkits show corresponding increases in complexity at those areas also. But be aware of those, and hopefully you still remember the phylogeny, and you can put these in their proper phylogenetic context.